So our last speaker before our panel discussion is Dr. Fiyinfolu Balogun, who I haven't also had the pleasure of meeting before. But he got his MD, PhD from University of Chicago, trained in medicine and HEMONC at NYU, and then moved a few blocks north to the Upper East Side, where he's assistant professor at Memorial Sloan Kettering in GI oncology. His presentation is entitled Improving Representation of Underrepresented Minority Patients in Pancreas Cancer Databases. So thank you for coming to participate.
Good morning. How's everyone doing? Good. All right, so fortunately, I think just like Dr. Gaddy said, I am following Dr. Hidalgo and Dr. Gaddy, and a lot of what I'm going to be talking about, they've already introduced it to you. So bam, 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 getting you closer to lunch. So I'm a GI medical oncologist, focused really primarily on colorectal and pancreatic cancer, see also neuroendocrine anal cancer, but research-wise, I tend to focus on pancreas and colorectal, with an emphasis on better understanding these diseases in understudied and underrepresented groups, both locally and globally. At the end of my talk, I would hope that, or beyond hoping, my intention is that you leave here having a better understanding of why better representation in genomic databases is important. We will touch base on, I'll touch on some of the precision oncology that Dr. Hidalgo talked about, and also clinical trial representation. So with pancreatic cancer, I'm focusing on pancreatic cancer. We're really we're in exciting times. So historically, for decades, we've just treated pancreatic cancer with chemotherapy, which I think of as this big dirty bomb that's poison you put in the system, kills the bad cells, hurts the good cells, and you hope the good cells recover better. But we're going into more precision oncology, more precision guided therapeutics, where we try to identify biomarkers, targets in the cancer cells that are not necessarily present or less so in the normal cells. That way, our drugs actually select them. So the first step of that is essentially the biomarker discovery. So in a population, and in this case, you look at a population, the patient population, pancreatic cancer patients, we try to identify this biomarker, this target that we see in the disease and not necessarily as much in the normal cells. The next step is, well, we found this target, let's try to develop drugs that will attack this target, and so we can kill the cancer cells. And then the final step after we do all that mixing in the lab is we take it to clinical trials and see all this exciting results we see in the lab, in mice, in animals. Does it translate to human beings? Do we see that we get that effect also? Precision oncology, while it's this new paradigm, is not so new anymore, because if we look at what's been happening with the precision oncology drugs, the first one was approved here, the FDA approved in 1998. So that's almost 30 years. It would definitely be old enough to drive, vote, do a lot of things by now. So this has been a while, but we can see from this, this works. So we can see that over time, we have really an explosion of the amount of approved drugs this is a graph that shows us approved drugs with precision oncology. And we see that in the last decade, really, we've really exploded in that regard. And almost half of all drugs now approved in oncology are precision oncology drugs. So precision oncology is catching up with chemotherapy and will be going ahead of chemotherapy, because we're really not designing any more chemotherapy drugs. It's just new precision oncology drugs. In pancreatic cancer, and here I just have a list of some of the ones that we currently use. And those with the asterisks are those that have actually been FDA approved. Drugs that are not FDA approved but guideline recommended are also on there. But these are all drugs that if someone has pancreatic cancer and we identify any of those markers, we can just off the shelf prescribe that for them. When we talk about pancreatic cancer, more than 90% of them are pancreatic ductal adenocarcinoma. So it's usually, we use those phrases or we use that term interchangeably, really. And it is, as you saw when Dr. Hidalgo showed it, it is really I don't know whether I'd say it's the deadliest or the most aggressive, but it's one of the worst solid cancers because it's the third highest cause of cancer mortalities, even though it's more like number 9 in terms of incidence. And as I mentioned, chemotherapy, even though we're making these advances, it's still the primary way by which we treat it systemically. Now, one of the proteins, genes that also you heard early on, and I'm emphasizing it here again, is KRAS. Now, KRAS is important in cancer in general, just because it is the most commonly altered gene that we see across all cancers. It drives the cancer. So when you have the alteration or mutation or dysfunction of the KRAS, it very often would lead to cancer. Because it is so common across all cancers, the most common, that makes it a really good target. If we can get a drug that would get drugs that would target it and treat it, we can treat a lot of cancers with this precision oncology. But had been mostly undruggable for a long time. It was just a really, really slippery thing for us to target. And for several reasons too. It doesn't have a good pocket for the drugs to sit in there. It's just really not so easy for us to have targeted it. And of note, the 3 most common cancers that have KRAS alterations are pancreas, lung, and colorectal, with pancreas being the highest. Now, all this changed a few years ago with KRAS G12C drugs moving quickly through here, because it was mentioned earlier on, we have sotorasib and adagrasib as the first ones that target the specific KRAS G12C mutation. So the KRAS can be altered in multiple different ways, and each one behaves differently. Now, this is good to remember because I'll circle back to it in a few slides. But we did see that this finally we're able to break through targeted in lung cancer. G12C is the most common alteration seen in lung cancer. So this was approved and it's being used to treat for lung cancer now. Fortunately, this also translated to pancreatic cancer. These drugs, we see that for KRAS G12C altered pancreatic cancer tumors, they also do respond well to these inhibitors. As Dr. Hidalgo said, it's good that they respond. We see here in the example I have here, the median progression-free survival, essentially how long we're able to control the cancer with this drug is just about 4 months in pancreatic cancer. So that's not a lot. It's not where we want to get to. But of note, about 80% of patients in these trials had really no other treatment options. So typically, survival after that we're thinking of about 3 months. So we're making progress because we essentially doubled that with this drug. This is exciting because KRAS, as I mentioned, is frequently altered in pancreatic cancer, the cancer that has the highest levels of percentage of alterations there. But the downside to it is it's also just this particular alteration, G12C, is just 1 to 2% in pancreatic cancer. So it's not moving the whole needle for us as much. But because that opened the doorway a little bit, we have a lot of drugs, several of which were shown earlier on, that are being designed, undergoing trials now, very likely to be approved soon, that are targeting different variants of KRAS KRAS alterations. Pancreatic cancer does have about 90% of pancreatic cancers have alterations in the KRAS, so being the most altered one. So it'll make a big difference when we can actually treat it in pancreatic cancer. And we do have the more common alterations. So remember, those alterations I mentioned are different, they behave differently, and we tend to have different drugs that target them. Overall, we see there's a different proportions, so G12D being the most common in pancreatic cancer. But we also did notice, it has been shown that when you look at different groups, so looking at Black and African-American patients with pancreatic cancer, those proportions of KRAS alterations are different from what we look at when we look overall. Now what we see obviously drives where we invest in terms of drug development or what we want to target. So when we look at subgroups, in this case, Black and African American patients, and see that this doesn't quite align or match up with what we see normally, then we're essentially, this group is not fully being considered in our drug development pathway. So for example, we see that the G12V is the most commonly altered alteration for Black and African American patients, but D is the one that's most common overall. And we know a lot of the drugs that have been developed that are allele specific, targeting specific alterations are targeting G12D. And recently published data did show that when you look even beyond just the Black and African American patients, when you look across the different groups, so this is using genetic ancestry, South Asian patients, African ancestry, East Asian and European, and we see that when we break down the pancreatic cancer alterations, this is the wild type KRAS, mutants KRAS. This is alterations in the same pathway as KRAS, but not the KRAS exactly, and these are the wild type. And we see that they're different proportions, and this is statistically different. So the biology of these cancers, this is telling us that the biology of these cancers could have some potential. There's a signal that there could be differences in different races and ethnicities. And this is not something we've been thinking of or really considered, and we've been doing much about in terms of our drug development or design. So now the question is, does it really matter what mutations, if we like this representation in drug development, does it really matter? If we design drugs based on one group, it's the same cancer, it's pancreatic cancer, same alterations, we can just use it across the board. And that's always a good question that comes up. And going back to what I talked about in terms of or what I presented early on, how our process of developing the biomarker, drug, and then testing it. The first step is really what gives us our knowledge database. So that biomarker discovery. And we see that these databases are essentially the information bank upon which our knowledge is. So whenever we want to understand the disease better, or we want to design drugs, we go into the database, and that information guides what we do. We see here just the US population demographics, white, Black, Asian, and Hispanic here. And then we look at the database representation of some examples of some of the large databases. And we see that it's disproportionate in terms of just the representation for our non-Hispanic white patients compared to, which is all above 80%. And we see it's representing about 60% of the overall population. Probability of developing cancer is about the same when you're looking at Black and non-Hispanic white, so that shouldn't affect this. But we see that overrepresentation here, and we see that in the other groups, the maximum is 10%. So we're thinking of much, much less representation. So this database that we're looking at, that we use to learn about this pancreatic cancer and to develop the drugs, there's a big underrepresentation of minority patients. Now, previously had been published has shown that when there's an underrepresentation, you actually lose some information, which we can all kind of imagine is the case. But it was shown that if you have less than mutations that are not very common, so 10% or less frequency, they become undetectable, they're undetectable in underrepresented minority patients. And this we see essentially representing different race ethnicities. On here, we see the surplus of samples versus deficiency of samples. And we see that in essentially all the underrepresented groups for different cancers, there's just a deficiency of samples. So we can surmise that we're missing detection of some of the important mutations that affect these groups because we just don't have enough representation of information from them. Now, the final part, which is what Dr. Gaddy also just talked about in terms of clinical trials. This is where, after we've developed the drugs, we want to make sure that it's effective and safe in the different groups. When you look at underrepresented minority patients in key clinical trials, they're not represented appropriately. And you can see here, just looking at, on the left side, I'm just looking at some of the data from pancreatic cancer trial enrollment, similar to what we had just seen from Dr. Gady, that we see here that in pancreatic clinical trials from 2005 to 2020, it's about 85% non-Hispanic white and much less for some of the other groups that we see here, 8.2%, 0.3 American Indian, Asian Pacific Islanders, 2.4%, and Hispanic, 8.5%. And all these, if you remember the numbers from before, in terms of just the demographics and the chance of getting cancer, they're significantly underrepresented in pancreas clinical trials. So not only are the drugs designed not including information that covers all these groups, but when we actually see how effective or test out how effective or safe the drugs are, we're not really including enough of these groups. And this is another key one too. This was just published a couple of years ago, that you look at the trials that we do a lot of clinical trials, but not all drugs would eventually get FDA approved. But even when you look at the key ones that led to FDA approval, we see that significant underrepresentation. So these are the trials for the drugs that we're using now. So the outcomes from these trials are the drugs that everyone uses. And we're still significantly underrepresenting a lot of the Black, especially Black and Hispanic patients. Now, we have advanced a lot in precision oncology, and we're seeing the benefits. Now, all these things that I've presented or shown to you is that this kind of comes down to that question I asked: is there a point, do we really lose anything from underrepresentation in the genomic databases? And just taking a look through, walking you through this is the x-axis, just shows us the different years. And this is really a graph that the y-axis is showing us the benefit you get from precision oncology. So as precision oncology has improved over the years, how are we benefiting? And this one just represents the average of everyone. When you look at, and this is focusing on ancestry, molecular ancestry, in a lot of the work, that I do, I'm going to introduce a couple of them at the end. We do look at molecular ancestry. I also look at self-reported risk because it does speak to different things. Molecular ancestry really does help us, I think, focuses more on the biology, or helps us more with understanding the biology. So, and that's very appropriate here. So when you look at patients of European ancestry, there's been a 9-fold increase between 2012 and 2023. So a 9-fold beneficial increase with precision oncology. So as we've been getting better, things have gotten better 9-fold. When you look at Asian patients, for East Asian patients, it's 8.5-fold. So not too bad either. When you look at South Asian patients, they have benefited too, but it's easily 2-fold less, or over 2-fold less, so 6.8-fold. So this, we're starting to see the difference, or the crack, that there's less benefit that some of these groups are getting. And you look at African patients of African ancestry, they've had just a 6-fold benefit over that same time too. So all of this is translating into the benefit or the effect that we're getting from precision oncology, this precision oncology revolution here. And this just gives us some examples of some of the mutations or some of the biomarkers and approvals that have happened along the way. So touching base really quickly on a couple of projects that we're working on. And before I pivot into that, some of the potential action steps, and this is, I think, one of the last slides that Dr. Gady presented, was things I wanted to do was further investigate these signals we're seeing. The pancreatic cancer, the biology of pancreatic cancer, is it different across different groups? And also to increase the genomic database representation. So some of the barriers we've seen, or we just saw and just talked about, there are a couple of them that we try to incorporate into our trial, but mostly improving access by patient navigation. In patient navigation, there are studies that have shown that patient navigation does help improve clinical trial enrollment when it's more intentional, whether it's racially concordant, especially culturally competent. navigation, it does make a significant difference there. So two studies that we're working on now that we're just excited about and getting started. One is the first one is PC Impact. I think I was hearing about, when I was hearing about the inception of this Healthcare Disparities Conference, that's kind of how we work through with this too. We started off thinking, well, the biology, we're seeing signals that the biology of pancreatic cancer in Black patients differs from white patients, but just signals. We don't have enough information to actually test this out. So we wanted to look more into that. And essentially two things we're doing is current data that exists, we're trying to clinically annotate it, get a better, so that we can actually ask those questions that these differences that we're seeing, are they significant biologically? But we also know we need more samples. So another aim of this project is to increase the number of patients with pancreatic cancer, Black patients with pancreatic cancer in the genomic databases, and that would help prospectively for further understanding these questions. And the second part really is, as we started to look into that, we realized that even in the patients of Asian ancestry and Hispanic patients, there were differences in biology, not quite the same as what we're seeing, or in different directions that we're seeing in the pancreatic cancer biology. And we wanted to expand our study to include other underrepresented groups. And this part of the project actually also includes or takes advantage of linguistic components targeted mostly at Spanish-speaking and Chinese-speaking patients, so that's the key difference between this and the other one, where we use that with navigation to improve the enrollment of patients in genomic databases. Thank you very much for your attention. I'm also a product of the WIN award, similar to Dr. Gaddy. It's been very helpful for me.